Thank you. And I would like to thank the organizers for this opportunity to participate in this very lovely conference. I am an alumna. I've been a Human Frontiers Fellow many years ago, and I have been interested in DNA replication ever since. And it is really a fantastic opportunity to sit down here and listen to the extraordinary progress being made by current fellows. So we are interested in genome duplication. And as we all know, genome duplication starts in mammalian cells from numerous genomic sites that are called replication origins, and that's the essence of the central feature of my talk. Genome duplication involves the chromatin unpacking, duplication, and repackaging, and it shares the template. It is an enzymatic process that shares the template with other processes that occur concomitantly on chromatin, including transcription, recombination, and repair. So anybody who had tried ever to untangle those, holiday lights can attest that this is not always a very easy process, and oftentimes things can go wrong. For example, when genome duplication occurs, too few or too many origins can be activated. Origins can be activated more than once during a single cell cycle. Replication can collide with transcription, and replication can stall a toxic impediment. So all those improper origin activation consequences are collectively known as replication stress, which is a phenomenon that is quite rare in somatic cells, but it is prevalent and often thought about as a hallmark of cancer, often underlying genomic instability in cancer cells. For us at the NCI, it is important because it's also a determinant of cancer chemosensitivity. We can exploit those replication stress pathways to design therapies. So what are we doing about trying to understand how cells regulate the replication origins? So the first thing we must know if we would like to understand replication origins is where they are mapped on mammalian chromosomes. And that was the subject of my Human Frontiers Fellowship when I first started. We designed a method to map replication origins, and then we have performed some analyses at a single gene level to map and understand the structure of those interesting genomic sites. Since I moved to the NCI and started my own group there, we continue to map replication origins, oftentimes in a whole genome level, and then we started to ask other questions. For example, when are these origins activated, since we know that not all the genome is duplicated at the same time during the mitotic S phase of the cell cycle. We also started to look specifically at protein DNA interactions that occur at replication origins to try to see how they are differentially regulated. And we started to ask a big question, which is why are some origins activated whereas others are not, since all of them basically bind the same enzymes that would lead to the unwinding and the catalysis of DNA synthesis. So how do we know why some are activated and some are not? We have deciphered some cases in which the protein DNA interactions that we have unraveled are important to prevent activation of too many origins, and that can occur either by activating too many origins one next to the other or by reactivating the same origin more than once. And this is what we are currently focusing on, trying to understand how cells prevent overreplication. Now, why this is important? This is important because if excess DNA synthesis has dire consequences, and some of those dire consequences are hallmarks of cancer cells, including the formation of numerous extrachromosomal DNA circles, transcription and replication collisions that lead to structure called R loops that are deleterious to cells, and shattered chromosomes. All those are hallmarks of replication stress, and cancers that can tolerate this excess replication depend on very distinct targetable DNA repair processes in order to sustain their proliferation, which means that they are often vulnerable to specific chemotherapeutic agents. And at the NCI, we're trying to understand these processes to try to predict which chemotherapeutic agents will be active in particular cancer types. So what did we learn? We first learned when we tried to look at replication origins that we do not have a very active consensus sequence that identifies all replication origins. We have different groups of replication origins. Some of them are active in most tissues, and these are associated with euchromatin histone modifications. And in other cases, we see that the origins are in cohorts that differ in their activation frequency between cell types, and tissue-specific origins are often associated with heterochromatin marks, such as specific histone methylations that we have discovered and reported. So we assume that those different origin cohorts that are specifically activated at different cell types coordinate genome duplication with transcription and with chromatin accessibility. But even though we are looking at different cell types, we know that within each cell type, there are still different groups of origins that are activated at different times during the S phase of the cell cycle. Some are activated very early during S phase, some are activated in the middle, some are activated later. So we are wondering if there are more subtypes of replication origins that we have not yet completely deciphered. And we started to look at proteins that are not the enzymes that are involved in the actual enzymatic process of DNA replication, but are binding groups of replication origins selectively. And we find several. Those are depicted here. And I would like to share three types of interactions, DNA-protein interactions at replication origins that regulate genome duplication that we are most excited about right now. The first type is interactions that prevent activating the same origins more than once. And these are mediated by proteins such as this one that is depicted here, RepID. The second type of interactions are interactions that prevent activated too many origins just next to each other. So they are actually negative interactions that bind replication origins in order to suppress the initiation from those origins. And the third type that I will not have time to describe in detail are interactions that coordinate DNA synthesis with transcription, chromatin condensation, and DNA repair. So let's start first with RepID, which is a protein that is associated with replication origins. What I'm showing here is a screenshot of a chromatin immunoprecipitation that delineates where RepID binds, and a screenshot of a nascent strand abundance assay that delineates where replication starts in the same mammalian cells. As we can see, those two sites are co-localized. And if we just line up all the sites that look like this in mammalian cells, we see that RepID is binding at the same locations as active replication origins. So what does it do? It is not participating directly in the catalysis of the DNA synthesis process. So what we found is that RepID binds to pre-replication complexes, and it does not play a direct role in the DNA replication itself. Conversely, it is recruiting a ubiquitin ligase called CRL4. And this ubiquitin ligase, CRL4, is binding pre-replication complexes that are associated erroneously with DNA that had already been replicated. So if those pre-replication complexes would assemble on chromatin that already had been replicated, DNA can re-replicate. And that is deleterious, because then we will have extra extra pieces of DNA, extra fragments of DNA that are in the cell, they can activate DNA damage responses. RepID recruited the CRL4 ubiquitin ligase, and that CRL4 ubiquitin ligase degrades the pre-replication complex, so basically blocks this interaction. And we have shown that RepID is indeed important to prevent re-replication at the cellular level. So what I'm showing here is a group of cancer cells that are stratified according to DNA content and according to the level of DNA synthesis. And RepID is prevented by bringing CAL4, prevents re-replication. In cells that are RepID null, on the other hand, we do see massive re-replication in the same cell type, and this is because RepID had not recruited the ubiquitin ligase in order to prevent the re-replication. So pre-replication complexes are recruited to chromatin that had already replicated, and then we see re-replication. Now, the second protein DNA interaction that I would like to mention involves SIRT1. SIRT1 is a protein deacetylase, primarily removing acetyl groups from nuclear proteins. It is a metabolic sensor because it senses the levels of NAD in the cell. SIRT1 binds replication origins, and this is exactly the same type of mapping that I have shown before for RepID. And we can see that the SIRT1 mutant that is mutated in the active site, so it does not deacetylate protein, can also bind DNA at the same location. Why this is important? This is important because there are other sequences that SIRT1 binds that we were puzzled about because they do not act as replication origins in cells that harbor the intact SIRT1. However, we found that they are active in the cells that are harboring the mutant SIRT1, or when SIRT1 is entirely absent, suggesting that those are origins that are dormant. They are not activated in the cells because SIRT1 is binding to that. So they are suppressed by SIRT1. What are these dormant origins? The dormant origins have been detected in cells that are subject to a DNA damage response. So when there are sequences that assemble pre-replication complexes, so they are ready to start replication, but normally they are simply not replicated. They are replicated passively from replication forks that start from their neighbor, the active baseline origins. However, when DNA damage occurs, oftentimes replication forks are stalled, and then the dormant origins can act as reservoirs, and they would initiate DNA replication to complete replication of the entire genome. So we were wondering if replication initiates more frequently when those dormant origins are activated, or does SIRT1 simply suppress a group of origins, but it is basically just decentralize the replication program. In order to do that, we used a single fiber analysis or DNA replication, also known as a DNA replication, replication assay by DNA combing. And in this assay, we label DNA fibers with nucleotides that we can detect sequentially with very specific antibodies. We spread them on microscope slides, and we look at the distance between the initiation sites, the distance between replication origins. So what we find here is that cells that contain intact SIRT1 have a longer distance between the replication origins on average when we map all the distances on our DNA fibers. Now, this means that the distances between replication origins are shorter without SIRT1, suggested that SIRT1 is suppressing dormant origins. So how does SIRT1 do that? We worked out the mechanism of SIRT1 suppression of dormant origins. And it turns out that, so in order to do that, we were looking for another protein that is binding specifically to dormant origins. And we found such proteins. Actually, we found a modification of the helicase. Helicases are the protein complexes that separate the two strands of DNA as a first step of DNA synthesis. This helicase variant, this helicase subpopulation is phosphorylated on a specific serine, in this case serine 108, and this serine is a target for a kinase, an ATR kinase, which is a DNA damage sensor. So when ATR phosphorylates serine 108 on this MCM2 protein, the helicase can be activated and replication can start at dormant origins. The ATR is brought to chromatin by TopBP1. It's a partner of ATR. And SIRT1 is removing an acetyl group from TopBP1. When the acetyl group is removed, TopBP1 no longer binds chromatin and does not recruit ATR, and hence we see that the dormant origin activation is being suppressed. So basically, we see that we have a balance between ATR activity and SIRT1 activity. So ATR is responding to DNA damage, or to signals that resemble DNA damage by basically trying to phosphorylate serine 108 on MCM2 and activate dormant origins. SIRT1 is deacetylating the ATR partner, TopBP1, and prevents this acetylation. And therefore, this is a way that the cells sense both metabolic activity and DNA damage signaling to regulate the number and the frequency of replication initiation sites. When the cells encounter either DNA damage, then ATR activity is increased, or when there is a higher metabolic activity, and then SIRT1 activity is enhanced. So in summary, what I've shown you is that we have found two pathways that prevent excess replication. The first pathway involved ubiquitin ligases, and ubiquitin ligases are preventing the whole replication complex from binding to DNA post-replication, and hence they prevent initiation of DNA replication on DNA that had already replicated, so preventing re-replication on the baseline origins. And SIRT1 and the components of the SIRT1 regulated pathway prevent initiation from dormant origins by basically countering superfluous ATR recruitments during normal replication when there is no perturbation and no DNA damage. And with that, I would like to thank the members of my group and the former members of my group, as well as my collaborators, and thank you for your attention. Happy to take questions.
Thank you.
I'm going to ask a question which is not scientific, but I was listening with amazement because your fellowship was in 1993, which is 30 years ago, HFSP, so you were one of the first generation of the... Could you share with us, if you look back, 30 years back, was that important, was that good, was that forgotten? What did you do for your fascinating scientific career?
So I was Human Frontiers Fellow at the Salk Institute. My mentor was Jeff Wall, who is just, I think he has his retirement party next month. We worked on genomic stability. We did not understand DNA replication. We did not understand where origins were, and we were just very interested in just learning where they are. And there were a lot of controversies whether replication origins exist or they don't exist, and is replication random or not random. So we needed a method. And we developed one thanks to the support, among other sources, of the Human Frontiers. And this basically helped me start my own group and started my own research. I stayed for a few years, then I moved, but that's basically the basis of what I have been doing since. So thank you.
Any questions? We just have time for one or two.
One more.
Yeah.
Oh, okay.
So does this hypothesis stand if there are secondary DNA structures formed during replication, like G-quadruplex or any other secondary structures?
Yes, so there are many of them, and part of I kind of mentioned briefly transcription replication collisions. This is a subject that we are very interested in, and basically what often happens is a situation whereby an RNA molecule is stuck between the two DNA molecules when they are opening up. This is deleterious. The cells have an army of RNases that are recognizing those structures, because those are formed very frequently, especially in regions that are very rapidly transcribed, such as the ribosomal DNA arrays. And when normally, it's kind of a matter of a balance between how many RNase H there are and how fast replication is going. When replication is perturbed, or when there is not enough RNase H to counter those R loops, then this is one of the situations whereby dormant origins are activated. So we actually have a follow-up project that I didn't talk about, where we looked specifically at those R loops in cells, in the rDNA region, in older people, younger people. It turns out that, unfortunately, yeah, I did this fellowship 30 years ago, so this is very depressing to think that older people are kind of not as good as younger in resolving those transcription replication conflicts. That's besides the point. We are still optimistic. RNase H inhibitors are in the clinic, in clinical development as anti-cancer agents, obviously, because the cells that have massive replication are kind of dependent on them in order to continue to proliferate. So, yeah.
Thank you.
Is there any other questions? No? Okay, thank you.
All right. Thank you.
Thank you.